Hello, everyone. Welcome to my talk. It's a great honor for me to present my last 4 years PhD work, which is entitled Investigating the Role of Genotype, Field Design, and Environment in Cereal Legume Intercropping System. So over the last 6 decades, crop yield has increased, except the recent stagnation in some part of the world. In this figure, we see that the crop yield increased since 1960 in Western Europe for many cereals and pulses and potatoes. However, due to global world population increment, there is also food demand, and it was forecasted that the food production in 2050 has to be increased by 26% in 2050 compared to the current level to fulfill the demand. However, the past crop yield increment was accompanied by a monoculture production system, which is also characterized by higher chemical inputs such as pesticide and fertilizer, which has a detrimental effects on environment and the marine ecosystem. For instance, the runoff of fertilizers and chemicals from agricultural and industrial resulted in the proliferation of algae, a phenomenon which is called a algae blooming around in Baltic Sea. This resulted in the bacteria which composes this algae consume a lot of oxygen and resulted in a dead zone for marine ecosystem or marine animals. Not only that, those chemicals has also a detrimental effect on terrestrial insects. This is a famous paper published 3 years ago, which stated that 70% of insect biomass declined over 27 years in protected areas. Pesticide has also a healthy risk to animals and even to humans. So if the same trend of crop production continued, it compromised the survival and well-being of the future generation. There are some initiatives. For example, European Commission proposed pesticide cut by 50% in 2030 under the strategy from farm to fork. But reducing chemical alone is not sufficient. So reducing chemical input requires a cropping system change, because it's not only enough to reduce chemical with the same crop production system. In this aspect, intercropping system is an alternative crop production system, which is cultivation of two or more species on the same land, and the species grow together for certain co-growth period. Several meta-analyses have shown that intercrop has high land use efficiency and improved resistance to pests, and also has higher yield stability compared to monocultures. It also allows lower inputs without compromising yield production. Nevertheless, the advantage of intercropping system is influenced by a complex set of factors, which include genotypes with other genotype interaction with environment and the management options. Therefore, it demands for further information regarding which species to combine, which genotypes of the species to combine, and which management strategy is good to increase land use efficiency by intercropping system. Here is where my PhD project comes in, which is categorized into three main objectives. So the first one is to understand genotype cropping system interaction by synthesizing available evidence and identifying the knowledge gaps in this aspect. Secondly, it's to test a new intercrop model by targeting a rapid or as a tool for rapid screening of possible management strategies for intercropping system. And thirdly, to apply this model to investigate resource acquisition and use efficiency in intercropping in combination with field experiment data. Let's see one by one. So the objective of the first chapter of this work is to investigate the effect of mixture genotype and identify knowledge gaps in genotype evaluation in intercropping system. For this, we conducted a meta-analysis which only considered cereals and legume intercropping system. And those studies which was conducted in field experiment and at least test two genotypes of at least one species. We extracted some data such as main interaction effects, that means the genotype cropping system interaction. Secondly, we extracted land equivalent ratio. This land equivalent ratio is an indices for land use efficiency. And please note that this is the term maybe I will frequently use in the rest of my presentation. LER is calculated as the sum of the yield of crop 1 in intercropping divided by the yield of crop 1 in monoculture plus the yield of crop 2 in intercrops divided by the yield of crop 2 in monoculture. And if land equivalent ratio is greater than 1, it indicates intercrops had high land use efficiency than monoculture. Let's see some of the results. So among the studies we consider for further analysis, 37 papers out of 49, which is 75% of the total, shows that there is a significant genotype crop system interaction effect. That means if you grow one genotype as a monoculture or in intercrop, it makes different responses. That means a genotype which are good or best in intercrops may not be the best in monoculture, or those genotypes which are good for monoculture are not necessarily good for intercropping. That means we need specific genotypes for intercropping, or we need specific plant traits to enhance land use efficiency in intercropping system. So the genotype choice affected the performance of intercropping system, and we went to look at what is the mechanism behind this, or which plant trait is responsible for such interactions. And we found that plant height was one of the often reported as a relevant traits, but other most important traits such as root trait or disease resistance were overlooked, or there is no studies in this aspect. If we look at the extracted land equivalent ratio from 35 studies, on average, intercrop have 26% higher land use efficiency compared to monocultures. But the distribution is that there are some points which are below 1, and there are several points which are above 1. And 1 is the point where we say intercropping is better or worse than monoculture. So the combination of the wrong combination of some genotype may result in less land use efficiency, or the combination of the genotypes which work together may result in higher land use efficiency. Therefore, the choice of genotype for intercropping system is crucial. But what we have found was there is a lack of data, especially on traits and genotype effects on intercropping system, which calls for further research efforts. But there is a challenge due to that we have a broad spectrum of species. So there are different species of the cereals and legumes, paleoclimatic conditions, and management options. Therefore, and it is difficult to test all of this and their interaction in the field experiment. This requires a modeling approach, particularly. Therefore, intercropping modeling is a promising tool to support the design of optimized crop mixtures for various conditions, which takes me to the next chapter. So the model I'm talking today, or the model I used in my PhD work, is a dynamic process post-crop model, which is a computation or simulation of crop growth development in relation to environmental conditions based on agronomic principles. And the model has some input data, and it considers plant processes, and then and it produces outputs. For example, the model inputs it includes weather data such as temperature, rainfall, and radiation, soil properties such as soil moisture content and soil nutrients, and also species properties. And then the model has some processes which include phenological development, light interception and utilization, and so on. Based on this, the model gives us the output such as biomass, yield, or water and nutrient uptake, which we further analysis. For example, this is a simple way to model or simulate daily biomass accumulation. And biomass over time is a function of radiation use efficiency, which is specific to crops, multiplied by the amount of intercepted radiation, which is further depending on the total incoming radiation, plant light extinction coefficient, and the leaf area index. So this is a simple example how we simulate biomass over time. Therefore, model can be assist or can be used as a virtual field to screen different genotypes, especially for intercropping system, which is complicated in field experiment and challenging. So we tested a new intercrop model for capturing a mixture effect, and I now describe some aspect of this model. So this model is implemented in Simplex platform, which is a scientific impact and modeling platform for advanced ecosystem management. So there are two components in this case. The below ground component and then above ground component. The above ground component is mainly for radiation competition. So the competition for radiation is mainly governed by plant height, leaf area index, and light extinction coefficient, while the below ground competition is mainly governed by root length, density, and rooting depth. So the uptake of the nutrient and the uptake of water is governed by the root density and rooting depths of each species. And so the two species have common soil, and then they compete for soil resources. But a model has to be first calibrated and tested with field experiment data before their application or before their use. And we used a PhenoRob mixture trials we have conducted in 2020 and 2021 in collaboration with a group of organic farming and agroecology of University of Bonn, which I'm very thankful for the data. And this experiment comprises two faba bean cultivars with 12 spring wheat cultivars. under two soil densities in two sites, which is Klein Altendorf, which is around 20 kilometers from Bonn, in 2 years, 2020 and 2021, and then Wiesengut only in 2020. Now I show you some results from model calibration. So we calibrated the model only in monocultures, and we let the model to simulate intercropping system. And the competition and complementarity that's implemented in the model should capture the mixture effects. This is a result from just grain yield for two soil density for each sites and observation. The line is observation, and the points are the lines are simulation, and the points are observation. And the second plot, the lower plot, is for plant height, and it seems that the model is well fit to the observation. And when we see the validation results, this is for absolute grain yield for two species, faba bean and springwheat in intercropping for three sites. The x-axis here is the environment, CKA 2020, CKA 2021, and Wiesengut 2020. And the blue one is simulated, and the green is the observation for both species. And it shows that the yield, or the grain yield, the simulated grain yield and observed grain yield is it's well fit. We also tested the model if it captures the mixture effect, and it is indeed the model captures a mixture effect on grain yield and biomass. And it's because our objective is also to enable the model, helps us to assist different management strategies. For example, the cultivars. We tested the model also if it captures the cultivar differences, and it's able to capture the difference between the spring wheat cultivars. Therefore, intercrop model is a promising tool for designing intercropping system and for further applications. Yeah. So experiments are limited in number of treatments, and especially in intercropping system, because of it necessitates also inclusion of monoculture, which resulted in cuts of dimensionality if we need to test different cultivars. But models can help to interpret experimental results in terms of crop growth, dynamics, and resource acquisition, for instance. I will now show you one example how we apply the model to understand resource use acquisition and interaction in spring wheat faba bean intercropping system. The objective of this work is to identify which resource underlay the performance of intercropping system, and is it also acquisition or use efficiency is primarily drive intercropping performance. Just when we see these figures, or different meta-analyses have shown that intercropping has high land use efficiency in monocultures compared to monocultures. For example, this is from our own study, from mixture trial, from simulation and observation in three environments. We see that even though the amount is, or the degree is different, but the land equivalent ratio is greater than 1 in all environments for both simulated and observation. What is the mechanism behind that? Why intercrops produce more yield compared to monocultures? What are the factors? Previous literature shows that it's attributed to complementary resource use, such as water, radiation, or nitrogen, but we don't know which resources are really main driving force, and we do not know also if it is is attributed to acquisition or use efficiency. Use efficiency in the sense that conversion efficiency, so the efficiency of converting the acquired resource to biomass and the grain yield. And therefore, the main question is it radiation, water, or soil nitrogen that enhances land use efficiency in intercropping? So we used intercrop model with field experiment data from 3 contrasting environments, so to identify which resource and which resources. And these environments are fortunately they are contrasting in those resources. For example, Campus Glen Altendorf 2020 is characterized by higher rainfall and high nitrogen, while Campus Glen Altendorf in 2020 was characterized by high nitrogen but lower precipitation. And Wiesengut, which is organically managed field experiment, is characterized by lower precipitation and lower initial soil nitrogen. So here we compare a resource acquisition efficiency of monocultures, the average of the 2 monocultures, against the intercrops. And to see some results in CKA 2021, which is characterized by higher nitrogen and higher precipitation, intercrops improved nitrogen acquisition by 17%, but it reduces nitrogen conversion efficiency or nitrogen use efficiency. Consequently, it also increases water use efficiency and radiation use efficiency to some extent. In CKA 2020, which is characterized by higher nitrogen and lower precipitation, intercropping relatively improved nitrogen acquisition, but it also reduced nitrogen use efficiency. But there are some extent of increase in water use efficiency and radiation use efficiency. In Wiesengut 2020, which is characterized by lower nitrogen and lower precipitation, nitrogen acquisition is increased by 22% in intercrops compared to monocultures, as well as the nitrogen use efficiency also increased by 13%. Similarly, water use efficiency and radiation use efficiency was also increased. But the most important message here is, while all of the environment have together, common thing together is that the nitrogen acquisition is enhanced in intercropping compared to monocultures. Additionally, intercropping improved resource use efficiency, specifically under resource limited areas, which is low nitrogen and precipitation, Wiesengut 2020. So this tendency that intercrop improve nitrogen acquisition mainly helps to reduce nitrogen inputs compared to monocultures. Beyond that, the model also helps us to understand the spatial and temporal complementary resource use. And here I show one example related to water uptake in 2020 and in 2021. First of all, under limited precipitation, which is CKA 2020, there is higher water uptake from deeper soil layer, from 90 to 120 centimeter deeper, compared to CKA 2021, which is relatively high precipitation. But when we compare the species, for example, faba bean in mixture acquired or uptake less water compared to faba bean in monocultures. However, spring wheat in mixture acquired higher water compared to spring wheat in monocultures. That means spring wheat is very competitive for water under water limited conditions. But in CKA 2021, remember that this is characterized by higher precipitation, there is no difference between the species water uptake between the two cropping system. For example, faba bean in mixture and faba bean in monoculture, almost the water uptake is similar. While the spring wheat water uptake is relatively, the spring wheat water uptake in mixture is relatively higher than that of spring wheat in monoculture. So there is no difference in water uptake from deeper soil, specifically in high precipitation areas. Furthermore, model can be, or this kind of model also can be also applied to identify beneficial trait and conditions that enhances the performance of spring wheat and faba bean intercropping system. Here we did a upscaling of intercropping model to regional level, which is in North Westphalian, Germany. We set up spring wheat faba bean intercrop model for 200 locations in NRW. And we combine or we tested different plant traits, which is a virtual isotypes and different nitrogen input. Those traits are root traits, plant height, and light extinction coefficient. So we have 3 traits and 1 management, which is nitrogen. And simply, it's like combining taller spring wheat with shorter faba bean, or shorter faba bean with taller spring wheat, or deep rooting spring wheat with shallow rooting faba bean, or shallow rooting spring wheat combined with deep rooting faba bean, and so on. And we simulate for 30 years from 1991 to 2020. And this combination resulted in 16,384 genotype by management combinations for each site. So each plant trait have 4 levels for each species and 4 levels of nitrogen for spring wheat. This resulted in 16,384 unique combination. And then for each 200 location, this is around 3 million data points. And then we calculate the mean land equivalent ratio over the 30 years for further analysis. Just to show some result, here is how intercrop perform in different soil and climate regions of North Westphalian. And as it's only default values, the important message here is that the land equivalent ratio is greater than 1 in all cases. And the root traits and interaction with the site and management condition determine the performance of intercropping, while the shoot trait were less importance. Specifically, intercrops use land more efficiently on when it is sandy soils, especially in these areas, with low nitrogen input when we combine a deeper rooting spring wheat with shallow rooting faba bean. This provide a guidance, especially for breeders, which trait to target, and also helps the farmers for site-specific variety selection and management by considering their specific production objectives. With this, I will bring my presentation to end. So a careful choice of genotypes in cereal legume intercropping system is crucial for enhancing land use efficiency. And the intercrop model we have developed and tested is able to simulate intercropping system by considering also differences in species and with cultivars, this is particularly important to test, to conduct a virtual experiment using this model. So the high land use efficiency of spring wheat faba bean intercropping system was mainly driven by enhancing nitrogen acquisitions. Therefore, designing a site-specific spring wheat fiber bean intercropping system enhances nitrogen availabilities and use efficiency. This helps to minimize nitrogen inputs. My PhD work also identified some research gap which has to be tackled in the future. First of all, identifying a crop traits, particularly a root trait that enhances complementarity in resource use efficiency is worthwhile to look. It's also important to consider a legume species or genotype with higher atmospheric nitrogen fixation capabilities to further reduce nitrogen input. And in scaling up and adoption of intercropping system, it is important to evaluate economic viability, and farmer adoption barriers of intercropping system is very important. Quantifying the impact of intercropping on other ecosystem services and biodiversity is also crucial to study. Most importantly, studying the mechanization challenges and solution for sowing and managing intercropping system, especially harvesting of intercropping system is what has to be tackled in future research. With this, I would like to thank people who have contributed to the success of this project, and I'm very thankful for your attentions, and looking forward to your questions and suggestions. Thank you.